using the same master stem cell bank across different types of tissues. Sorry, just here where we are.
Yeah, it's a little confusing here.
What you run the risk of is that you run into a problem in one study. Say you're making mesenchymal stem cells. You have, something comes up in that study that is a problem where the FDA can't discern whether it's because of the stem cell line or because of the tissue you made.
I see.
And we worry that their response may be stop using the embryonic stem cell line in everything until we figure it out. One of the ways to mitigate that risk. What's it here?
Sure.
One of the ways to mitigate that risk is to have a different stem cell bank for each type of transplantable tissue you want to make. And that way, if there's a problem that comes up in one program, the other programs have a firewall because they have a different stem cell line. So that has been, one of the issues we've been sorting through as to whether we want to make additional lines and how many we want to make. The line that we used in the RPE study doesn't include anything other than human products, but when it was derived, it was put in culture with mouse feeder cells, which is a typical thing to have done then. It means that the regulation of it is a little more complicated, only because it means every patient you have to get a blood sample and keep it for the rest of their life. Whereas if we derived a new cell line, like we've talked about NED7, one of the advantages to a new cell line is we can make it without feeder cells or use human feeder cells and avoid that whole issue altogether. So there's no definitive answer in terms of what we're going to do, but those are the types of considerations we have as we think about the opportunities and decisions in terms of future cell lines.
Okay. Can I ask for phase 2, will each injection be DSMB approved just as the previous phase 1?
So right now the phase 1 study has a lot of DSMB oversight. It's not quite every injection, but it's enough that it makes it cumbersome.
Right.
So phase 2, our expectation is that there'll be the DSMB review will be farther apart in terms of being able to treat more patients before DSMB review is required. I think we're also probably going to restructure the DSMB in a way that is able to handle the volume of patients we're anticipating for phase 2. Right now we have a lot of academicians who are wonderful, but we're asking them to take a lot of time out of their otherwise busy schedules to be looking at this data over and over again every couple of months.
And do you have about a number? How many patients will be in phase 2?
No, not yet. That's still that's still a hotly debated topic and one that we're actually requesting guidance from. We have a proposal, but it's not something I can say publicly.
Understood.
But we are looking to the MHRA and the FDA to review the proposal and provide some guidance as to whether they think that's reasonable or not.
Okay. Has ACT submitted to the FDA the paperwork for phase 2? I'm just reading off that list.
No, not yet.
I think you might have. Yeah. Okay.
So we're still in the design phase, and just getting into the kind of direct discussion phase with the FDA, but not something where we have filed the paperwork necessary to get approval of the phase 2 design.
Okay. Yeah. And are we going to be separating AMD and SMD in phase 2? Or?
I think so. I think the strategy is going to be a little bit different. At least that's the way it's currently being proposed.
Okay.
In part because the diseases have different demographics. Stargardt's, because it's an orphan indication, also has a whole different set of incentives that we get to take advantage of in a way that it's not clear that we can move in the same way with dry AMD for now.
This I'm going to just read verbatim. I hope you don't mind.
Yep.
Yeah. Matt, we've heard various statements from the company regarding when top-line data will be released. For example, there was a PR last year that indicated top-line data. Well, you saw the question, right?
Yeah. So, Here, Simon, here's the only way I can answer that right now. The data that we've been seeing has us and our investigators excited enough that they don't want to do anything that would prevent the data from being published in a very high impact journal. And so the negotiation right now with all of the clinicians who are co-authors on this paper is, what can we say today that won't jeopardize a journal picking up the paper, the type of journal that we want to be in.
I don't want to interrupt.
Yeah, being interviewed.
He's an important guy.
That's true.
I think
Are you going to race out to the airport as soon as you're done?
No, I've got a 2:30 and then a 3, and then I'm done at 3:30. But then I got to head downtown. I was going to try to catch the 5 o'clock train. I have a little time then. I'm the only one here.
I might have time at. I have a meeting at 3, which is probably also over at 3:30ish. So maybe we could just grab 15 minutes.
Yeah, you want to meet on the 3rd floor at the beverages, or do you want to come down here?
Do I need a pass? If I need a pass,
Oh no, I'll meet you down here.
Okay.
Why don't I meet you at the clock tower at 3:30?
Sounds good. If for some reason my meeting runs late or yours, we'll just play it by ear.
Okay.
Okay, good.
Thanks,
Sorry to
Michael.
interrupt.
Yeah, so I'm going to try and edit as much as I can.
No, don't worry. And email me if you want me to clarify anything.
Okay, sure. Actually, could I ask you, could I get the presentation? Because I think there were some new slides I've never seen before.
Yeah. So I'll send you a PDF of that because it'll be loaded up on the website soon. But I'll give you a PDF you can share with the folks at.
Sounds good.
Just tell them. I'm doing it as a favor to just 22. Tell them.
Okay,
so that John feels good.
Right. Thank you. Let's see. Okay. Will ACTP announcing the new CEO in next month or two?
I doubt it.
Okay.
I think that Gary did a wonderful job dealing with a lot of the financial issues that were left behind for him. I think even he foresaw, though, that where we are as a company necessitates a CEO with a tremendous amount of experience in terms of taking products the rest of the way through the clinic, facilitating large-scale deals, potentially with co-marketing rights, which means that we'd have the ability to sell the product ourselves. And so, given where we are, given the data that we could share with the CEO candidate in confidence right now, we feel like we can really get a high caliber person, but we're just at the beginning of that search. So.
Well, I'm glad to hear that at least he left it in good terms.
Yeah, no, I loved Gary. I emailed him today saying how much I missed seeing him here. So.
I know he got a lot of flack on the site and everywhere, really, in Google as well. But,
This company wouldn't be here today if it weren't for his efforts.
I see.
I mean,
That's
I don't think a traditional biotech company could have saved ACT with the financial situation that Gary walked into when he first became the interim CEO and then the CEO. It took his skill set, I think, ultimately to work through, they may be unpalatable solutions that kept the company intact and going.
Right.
So.
Someone asked about the employment agreement for Dr. Lanza.
I don't have any information on that. I know Bob's happy. I talked to him, multiple times already today, and I think today's his birthday, by the way.
Oh yes, actually, I wanted to say to pass our well wishes.
I will. I'll let him know that you guys say that. So,
actually, that was the first thing I wrote, but I forgot about it.
Yeah. So Bob doesn't celebrate his birthday. He was actually calling me. I've been working on this manuscript with him, so he was calling me today, asking me to redo a couple of the statistical analysis for it. But he's very happy. So and comfortable and confident. So I don't know the back office reason why the deal hasn't been signed, if it hasn't been, or if it has been signed, why it hasn't been announced. I don't know the status. I just know that I'm not concerned that
Okay.
Bob's going to leave. I think that ACT is Bob and Bob Lanza is ACT.
Okay. There's a question about the time frame for reverse split and uplist. Can we give it a ballpark?
Well, the authorization we have from the last shareholder meeting requires the reverse split occur by September 31st if it's going to occur at all,
I see.
without another authorization. So that's our target, and it also goes to why a high impact journal publication can be so important. The timeline for all of this, I don't know exactly when we would look to the reverse split in the uplisting to NASDAQ, but it would be nice to have it tied in in a way with the data being released in a meaningful format to really attract the interest of institutional investors who would then hold the stock long. And I think that helps every retail shareholder out there to have a solid base of support like that.
Okay. And but then you don't have any idea which comes first, the top line data or the first capital raise. That's not something that
I don't. Yeah.
Right.
I'm not. That's been worked out. I mean, what I can tell you is Mike Heffernan, who's now the chairman of the board, and Ted Miles have been working closely together through that whole strategy. So we're getting a tremendous amount of mind share from Mike now as well. But I'm not in those conversations.
Understood. Yeah. And can you share some progress on the vet trials, the injections at Tufts?
Yeah. So we've. I don't know how many of each of the different animals that we have the INAD approval to test in, but I know that we've been already working on shipping cells to the folks at Tufts, and they've been involved in the injections. One thing I would maybe ask to be passed along to Investor Stem Cell is that people respect the relationships that we have with collaborators, and when they call Tufts, which has happened, it actually puts those relationships in jeopardy. And I know how passionate our investors are, and how much they want to know what we're doing.
Yeah.
And believe me, I wish we could tell everything, but that's not the way to get the information, because at the end of the day, it came close to unraveling that relationship.
I see.
So what I'm saying, this is a long-winded way of saying, whatever I'm telling you now, there's not, just ask people not to try to dig any deeper at Tufts. Just let it'll develop. I think it'll produce some exciting results, and everybody will be happy, because hopefully it'll have a very meaningful impact on their share prices.
Right. Okay. Has the company made any progress in partnering the non-core programs?
We've had a lot of discussion. We actually even have some evaluation relationships going on right now. But a lot of what we have that's preclinical is still very early stage. So I think it's going to, requiring somebody who is willing to take a lot of risk early on to us, which means not a lot of value if we do a deal like that. Or that we have developed the data package a little further, which is what we're actively working on in each of these areas, and then it becomes more attractive to partner, and also the value goes up. So, a lot of interest, and I think it just remains to be seen as to when people come over the hump and want to do the deal, and whether it's a deal that we want to do at that point.
I see. I saw on the slide that there were 33 patients. So are we going to have the full 35 done before the submitting the top line data?
I think with all that remains in the trials at this point are cohort 4, which I think that we may redesign cohort 4 a bit, so I don't know when that will start. And then the last patient in each of cohort 2A, right? I anticipate those last 2 patients will be treated in the next weeks to month. I don't know for sure. But since I'm not completely sure as to what will finally resolve itself around top line data release and the whole trajectory of the scientific journal article. I don't know how those will line up